Firstly, a warm welcome to this video podcast of the Wits Journal of Clinical Medicine. I'm Pravin Manga, editor of the journal. For today's video podcast, I'm honored to have Professor Lynn Morris here with me today. Lynn is Deputy Vice Chancellor of Research and Innovation at the University of Witwatersrand in Johannesburg, South Africa. She's also Director of the Antibody Immunity Research Unit based at the NICD. And her major research interests lie with HIV over the last 28 years, and she has made major contributions to the understanding of the antibody response to HIV, and hence her major interests lie in development of an effective HIV vaccine. So as a background, Lynn, for our viewers, as of 2020, there are about 38 million people living with HIV in this world. And not so good for us, 68% of these live in sub-Saharan Africa. And in 2020 alone, there were 1.5 million new infections with close to 700,000 AIDS-related deaths. Unfortunately, South Africa has the highest prevalence of HIV in the world, and almost 7.8 million of our population, which is almost 13%, as stated in 2020. So that is a background. An effective HIV vaccine would certainly be a game changer for us. So here's a point, Lynn, that I want you to elaborate on. You agree that the research in the development of the HIV vaccine has in fact been a major springboard for the rapid development of vaccines against COVID-19, and the scientific world has been able to deliver so many effective vaccines for COVID-19 in a short space of time. So why have we not found the same for an effective vaccine for HIV?
Well, first of all, thanks very much for this opportunity to talk about HIV, because I think one of the concerns is that we have forgotten about HIV. We certainly need to remember that it still is a major killer of people in this country and of the world. And you're right, we're not going to get on top of HIV until we get a vaccine. So it really needs to remain a priority. So in some ways, you know, the COVID epidemic has sort of given people the impression that making a vaccine is easy. Because we had a number of effective vaccines in less than a year of having identified SARS-CoV-2. And that's just remarkable. I mean, it's a real triumph of science that we were able to do that. And they're highly, highly effective, as we know, particularly against severe disease and death. As we do know, they are less effective against some of the variants, and so they're not perfect. But of course, really what we want vaccines to do is to prevent severe disease and death. So from that, from that standpoint, they're still highly effective. But work is continuing. So how did we make a SARS-CoV-2 vaccine so quickly? Well, actually the answer is because of the years or decades in fact, of research and effort and funding that went into trying to make an HIV vaccine. So, so HIV is a very different virus to SARS-CoV-2. HIV is very, very mutable. In other words, it changes very, very rapidly. And that's really been, it's a moving target. SARS-CoV-2 in the beginning was, it looked like an easy target, but of course now we're seeing the variants are coming up. And so but certainly, HIV is far more variable than SARS-CoV-2. But actually the biggest problem with HIV is that it integrates into the human DNA. So it's a chronic infection. And it means that it's very hard to get rid of. Once it becomes an integral part of someone's DNA, it's impossible to dislodge. We haven't been able to cure HIV. Whereas SARS-CoV-2, it's an acute infection. The immune system clears the virus. Most people, don't have long-term effects, as we know. I mean, I know there are some, there is, the long COVID that occurs in some people, but for the most part, it is an acute infection. So already that's a model to tell you that it's much harder for your immune system to deal with HIV. And then the third thing is that the envelope protein, so that's the protein on the outside of the virus, which is really the equivalent protein on SARS-CoV-2 is called the spike protein. And I think, everybody now knows about spike because all of the vaccines are basically spike derived. So the HIV envelope or spike equivalent is a much more complex protein, and it has these convoluted, what are called epitopes, or the targets for antibodies. And so it's just inherently a more tricky protein, to work with and to deal with. But I think there's going to be some exciting news on that front too. So, because of what we've learned from SARS. So yeah, it's as I say, it's I've been working on an HIV vaccine for decades, and not making much headway, if one's really honest about it. There have been some small signs of progress in the HIV field, but overall it's been a disappointing field, because it's a tough nut to crack, HIV. It's not an easy virus to contend with. But what I can say is that that it wasn't, certainly not wasted effort, because that all went towards helping us get a SARS-CoV-2 vaccine quickly.
Okay. So now that you've highlighted that HIV in fact is a chronic disorder, with the HIV virus in fact becoming part of the host genome, what are some of the vaccine strategies that have been tried in the past, and which of these have been successful? We know that the Thai trial, the RV144, was the first one, I think it was published in 2009, to show some degree of HIV vaccine efficacy. Tell us a bit about that initial trial, albeit only partially successful, and where has this trial taken us to the next step?
So thanks for that. So because it is, this virus integrates, the focus has very much been on antibodies, because we need to block all viral particles from infecting, so from getting across the mucosa and infecting cells. So because once the virus integrates into a cell, as I say, it's almost impossible to dislodge. So the focus has very much been on envelope and been on neutralizing antibodies. But even in natural infection, so people who have HIV, even naturally, very few of them make those good antibodies. And again, that's very different to SARS-CoV-2, where everybody makes good antibodies to SARS-CoV-2, either following infection or following vaccination. So again, it tells you, about the complexity as well of the envelope glycoprotein. But it also tells you something about what HIV does to the immune system, that kind of inhibits, our body to make these good, these good antibodies. So that's always been the aim of HIV vaccines, is to try and make good antibodies. But of course, antibodies rely on T cells. They T cell help. So it isn't just about the antibodies. We do need a good stimulus for both arms of the immune response, so that we do get good memory B cells that will continue to make, the neutralizing antibodies. So the trial that you referred to, the RV144 trial that was done in Thailand, which showed a moderate efficacy, and it really was a little blip. In fact, to everybody's surprise, the correlate of protection in that trial was not neutralizing antibodies. It was antibodies it was an antibody correlate, but it was binding antibodies. And so we don't know exactly, what the, how this worked. And of course, it's a surrogate. It's not a direct correlate. But we think it's more to do with the Fc effector function of antibodies. So not the neutralization part, but the part that engages the cellular arm of the immune response. So the more innate immune response. So bringing in the killer T cells, the phagocytosis as well. And in fact, in some animal studies, phagocytosis has been shown to be, a potential correlate as well. So actually, what the RV144 trial showed us is actually, that it's a different functional aspect of antibodies that were, were working there. And we know from animal experiments, that those antibodies are just not as effective as neutralizing antibodies. And so, it really, and I should add as well that we've had, 2 subsequent trials to try and replicate, that result. We had a big trial here in South Africa, the HVTN702 trial, to try and replicate the trial that was done in Thailand. And very disappointingly, we didn't even see a small signal. And so, you know, the sort of feeling is that we really need to go back to the drawing board and focus again on neutralizing antibodies. And I think also, you know, SARS-CoV-2 has shown us, we know that neutralizing antibodies are a correlate, you know, for COVID vaccines. And so really we're going back to, you know, to trying to re-engineer the envelope glycoprotein, try some of these newer platforms that have been so successful with COVID to really see if we can elicit neutralizing antibodies.
So, I mean, from my reading, you can target a vaccine to potentially protect individuals at 3 levels. So basically, sort of what immunogens you believe are the most promising, like targeting the envelope protein, and especially in HIV. I mean, do we target the conserved area, which are more predominant in the variants of HIV? Does this immunogen produce both B cells as well as T cells? And in fact, then what sort of entry do you get into the human, whether it's vector-based or a messenger RNA-based?
Mm-hmm. Yeah, so one of the, you know, the exciting areas that you know, we've been involved in is trying to identify what these targets are on the HIV envelope, because you really want something that the virus needs. And so if you have an antibody against it, you basically debilitate the virus. And so there've been a number of very nice basic, you know, science studies looking at these individuals who do develop these broadly neutralizing antibodies that are very potent against multiple different HIV strains. And there's been a lot of work going into characterizing what are called, you know, these epitopes or these targets. So we've identified about 5 or 6 of these very vulnerable spots on the HIV envelope. And those are the sites you want to target. So the one that there's been a lot of work done on, and it's very easy to understand why it's so important, is the site on the virus that binds to CD4. So if the virus cannot bind to CD4 on the cells, it won't be able to enter and it won't be able to infect. So if you block the CD4 binding site on the HIV envelope, you basically neutralize the virus and prevent infection. So characterizing those, you know, those epitopes and those sites has been it's been a major effort from multiple groups and just very elegant work to really understand, you know, how those sites all work. So we've got a pretty good handle, you know, on the envelope, not only the sequence diversity but also the structural diversity of envelope, because of course, you know, the heterogeneity or the diversity you see in envelope impacts, of course, on its structure. But we've got a pretty good idea now of what we need to make this envelope look like it should do on the virus, because that's really what we're trying to trick the immune system into seeing, is something that looks like the virus. So there's been, you know, and actually another major advance is when people were able to design through the structure-based, you know, approach to designing this complex protein in the lab and manufacturing it, and then, you know, using that as an immunogen. So the problem with that, of course, is that once it goes into someone's body, of course, it starts disintegrating and, you know, different epitopes get presented. And so in some ways, it's almost a little bit of a black box. You know, you've created this beautiful protein. It looks perfect under, you know, the electron microscope. But once it goes into the human body, whether it's still presenting the right epitopes, we don't know. So this is where I think the mRNA technology is really exciting. So as we all saw from COVID, the mRNA vaccines have been by far the most successful. You know, really, I mean, actually, you know, they exceeded everybody's expectations, even Tony Fauci's. You know, right in the beginning, you know, everybody was talking, if we had a vaccine that was 50% effective against COVID, we'd be excited. And then, you know, the number went up a bit. And in fact, we know now, certainly against the original variants, you know, these vaccines were in excess of 95%. You know, effective, and they were effective against transmission and disease, which is also important. So the mRNA technology is really exciting. And to be honest, I think it's changed vaccinology forever, because I think everybody's going to try and make vaccines using mRNA going forward. And there are a number of reasons for that. The one is that they're much cheaper to produce, and they're much more adaptable. So it's, the vector-based vaccines or the protein vaccines, require, manufacturing capabilities. Whereas mRNA, actually what you're doing is you're outsourcing the production of the vaccine to the human body, because once the mRNA goes into the body starts, using that as its template and making the spike protein from the mRNA. And so actually our own bodies become the factories for making the vaccine. So, and particularly when you think about the importance of conformation, of presenting the envelope protein to the immune system in the way that it would normally see on a virus, you can see how the mRNA offers a lot of advantages for HIV, because we're now making the envelope protein in the body, so the immune system sees it immediately as it gets expressed on the cell in the right conformation, and hopefully that will improve the immunogenicity of the vaccine. So this is, all still speculation. That if you just have the right structure, you'll get the right immune response. And as we all know, it's never really that simple. But this is being tested now. So there is an mRNA vaccine for HIV, and the clinical trials have just started. So there's a lot of excitement about whether what we've learned from COVID is really going to help us to make an HIV vaccine.
So there was a publication in Nature Medicine about the mRNA vaccines in rhesus macaques that showed some degree of efficacy as well.
That's exactly right, and so that's where a lot of the enthusiasm is coming from. So in an animal model, we can see that there is already an indication that the animals are making these broadly neutralizing antibodies following so this is also, they put the mRNA into what's called a VLP, a virus-like particle. Mm-hmm. So the envelope protein gets expressed on the outside of this particle. So it really looks like, an HIV, a real HIV envelope on the outside of a particle. So it's being presented in the right way. So, but as I said earlier, even people who are infected with HIV don't always make these antibodies. But of course, HIV is also undermining the immune system at the same time that it's presenting, antigens or, to the immune system. So obviously a vaccine wouldn't be doing that. It's just presenting, the immunogen without undermining the T cell help and all the other things that happen in HIV and the dysfunction of the immune system. So yes, so watch this space. We still don't understand a lot about mRNA vaccines. We don't even really know which cells it gets into. There's some speculation that it seems to get into the follicular dendritic cells and lymph nodes, and that's obviously where immune responses start in germinal centers, and that somehow, it's getting right to the sort of site of action, and that helps with its immunogenicity. So there's still a lot to be learned, but hopefully the results from this, this trial that's just started will in about 18 months.
Is South Africa involved in this study?
No, the study is it's happening in the US. It's a very small phase 1 study. So these initial studies are really largely about safety. And I should say, and of course we know now because billions of people have received mRNA vaccines, that they're very safe. So I would imagine that phase of the trial should go pretty quickly. But the critical thing is going to be whether it elicits the right kinds of antibodies.
So the hope is if it does elicit the right kind of antibody response, the efficacy of the vaccine will just go up markedly.
Indeed.
Hopefully.
So there's another complicated thing about HIV. So because as I said, it's at every point, I think, every turn, HIV has presented new challenges. So one of the other very important pieces of information that we've got over the last year with HIV is the levels of antibodies that we need to protect us against infection. And this is actually the results from a trial that was completed last year called the AMP trial, the Antibody Mediated Prevention Trial. And this is where people were given VRC01, which is an antibody to the CD4 binding site that I mentioned earlier. And so this is passive infusion of antibodies, so passive immunity, which again, as we know from COVID, there were a number of antibodies that have been tested. They haven't been widely used, and I think that's really because we have such good vaccines, so there's been no need. But the idea of having a biologic, it's a biological drug that you could immediately give to somebody when they've been exposed, and it just mops up. It's instant immunity, in fact. You can just inject antibodies into people, and those antibodies mop up the virus immediately. So that's a very appealing, thing. Particularly, antibodies, I think, could have a real role in outbreak situations, for instance, where there isn't time for immunity to develop. So, but from the HIV point of view, it was really just a proof of concept. We wanted to prove that you could protect someone from HIV using an antibody. And so this trial, which was really, a fantastic trial that was done, was run by the HVTN, the HIV Vaccine Trials Network, and we were a major part of that trial. So the trial was a trial done in Africa and a trial done in the US, and the results, as I said, were released last year. So overall, the trial did not show efficacy. And the reason for that is because VRC01, the antibody, is not the best antibody. It was the first antibody, and this was a proof of concept study. It was not a proof of product. So we weren't proving to show that VRC01 was going to, become a product, but actually that it works. And indeed it did work, because when we did the analysis of the viruses that were infecting people in this trial, so it was a phase 2b trial. It was an efficacy trial. So there were in fact 3 groups. There was a placebo group, and there were 2 dose groups: 10 milligrams per kilogram and 30 milligrams per kilogram. But when we analyzed the breakthrough viruses in this trial, what we noticed is that all of the sensitive viruses were blocked. So that the antibody basically prevented infection of viruses that were very sensitive to the antibody. If the antibody had been more potent, it would have blocked more viruses. And so it's really an issue of we need more potent antibodies. And the good thing is we have more potent antibodies. And so, this passive approach to preventing HIV infection is continuing. And also with combinations of antibodies.
Like COVID.
Yes, indeed.
Indeed, available.
That's right. So, but the critical thing that's come out of the AMP trial that I think has been quite sobering is that the level of antibody that you need to protect from HIV is quite high, because you're trying to prevent every single virus particle from getting across the mucosa and infecting people. Now, most vaccines don't work that way. Most vaccines actually, as we see now with COVID as well, are preventing, there's still an infection, and they actually prevent the downstream, effects of the infection rather than blocking all of the initial particles. And in fact, for most vaccines, they work post-acquisition. And so what we're asking an HIV vaccine to do is to block every single infection, and that's why you need very high levels. So again, it is a bit of a wake-up call for HIV, but it's certainly not deterred us. We feel that this, this is something that's definitely worth pursuing. And the passive immunization is also something that people are seriously considering. If we can get a triple combination of antibodies that is highly, highly effective, and we now can engineer antibodies to have very long half-lives, up to 6 months.
Sure.
So if it's an injection every 6 months for people at high risk of HIV infection, and as you know, some of our communities, young women, more than 50% of them are HIV positive. This is a crisis, actually. But so if we can give them an injection every 6 months during that time where they're most at risk, that could have a major impact. So I guess we're always rethinking things, and But I think, the research needs to continue because you also never know what's going to come up.
Because HIV is such a huge problem in this country, what about the levels of funding that we have from government? Do you think we need much more support?
Funding is always, of course, it's always an issue because research is expensive, it's a technology-driven thing, and so one does need, to be investing large sums of money. But, I think it's also about using the money we have and using it well and using it, effectively and carefully. I should say, that overall, we've been very well funded for HIV work because it is a global effort, and there has been a lot of investment from, the National Institutes of Health through the HVTN, through the Gates Foundation, the Wellcome Trust, and also from our own government. There's been a lot of investment from the Department of Science and Innovation. And of course, the Medical Research Council really prioritizes, HIV vaccines and vaccines in general, because we know that vaccines are the best way to get on top of, these public health problems.
Now that we're developing some kind of vaccine manufacturing capacity, you think that this will be possible for HIV vaccines as well?
Yeah, so I think that's been another very positive development. Obviously COVID's been, a devastating thing globally, and obviously in South Africa too. But I think given that we were always on the back foot in terms of, access to vaccines, and it wasn't only vaccines, it was also access to laboratory reagents and resources to tackle the pandemic. So one of the positive things I think is highlighted how we really need to be much more self-sufficient. And there's been a lot of very exciting announcements. The mRNA hub, that the WHO and the French government have put together, and actually, Wits is heavily involved in that, together with Stellenbosch and other campuses, other universities. So, I think there's a lot of excitement around just our ability now to develop our own, manufacturing plants, and it's not just for COVID. In fact, they may be too late for COVID actually, yes. But it's for future, because there's no doubt there are going to be more viruses that cause epidemics, because viruses are very good at doing that. So I think we'll be better prepared in future. But it is also a sobering thing to think about our history. We used to have very good vaccine manufacturing in this country, and unfortunately we lost that capacity, largely because it was cheaper to just import them. So I think we need to think more strategically about, it's not just about the cost, it is actually about having the technological ability to to solve our own problems.
So thank you, Lynn. You've provided some great insights for the viewers into the biology, strategies, funding initiatives, and of the promise one day of developing a highly successful HIV vaccine. Thank you very much.
Well, thank you very much for this opportunity. Thank you.